the recessive allele is still there. It's just that it is not expressing. Okay, now you know that. Okay, after they form the gamete through what, meiosis. Then you have gametes that you have 50% of the gamete that has the big P, the purple allele. You have 50% of the gamete that has the white allele. Then what if you cross F1 generation with each other? So again, this is fertilization. Okay, I just put fert. And then, so how do we determine 3:1 phenotypic ratio? I hope you still remember this term, 3:1 phenotypic ratio. So let's say, okay, traditionally we put the sperm on top, and then we put the eggs on the side. As I said, that's approximately so you got 50% of the sperm that has the purple big P, and 50% of the sperm you have a small p. Similarly, you have 50% of the eggs that has the big P, 50% of the eggs you have small p. And then you just fill out, we call this Punnett square. This is what we call Punnett square. So you just copy. For example, in here, P, and then you see the small p here. You put the small p, but in convention, is that we put the dominant allele first. So we put the big P in front of small p. Similarly, we put the dominant allele first. So the big P is here. The recessive allele follow. And then this one is small p, small p. So small p, small p. So that's how we fill out the Punnett square. And as you can see that if it is big P, of course we get the purple flower. If it is the I'm talking about this one, big P, we get the small purple flower. If we get the big P, small p, which is the heterozygous dominant, just like the F1 generation, then we get the purple flower. We also get this big P, small p from the similar to the F1 generation. It's also purple flower. And then the small p here, it is similar to the parent generation. And then we know that it is the white flower. So the phenotypic ratio is 3 purple, 1 white. That's just something that I told you to memorize, 3:1 phenotypic ratio. Now we have a new term. Genotypic ratio. Genotypic ratio. What is the genotypic ratio? The genotype that we are talking about is the big P, small p, and small p. So we count from the Punnett square. How many of them we got? So we have 1 big P, 2 small p, 2 heterozygous dominant. This is 1, this is 2. So we got 2 heterozygous dominant, and then 1 homozygous recessive. This is called homozygous recessive. So that's why the genotypic ratio is 1:1. I also want you to memorize this. So phenotypic ratio is 3 purple, 1 white, but the genotypic ratio is 1:1. So I want you to memorize the 2 ratio. Okay, Punnett square. Okay. So Punnett square gives you the 4 possible combination, resulting in 4 possible offspring. Okay, in the F2 generation. Each square represents equal probability. Equal probability, okay? So I highlighted the definition, okay? Physical appearance is the phenotype. Genetic makeup is the genotype. Okay, do not mix up the two terms. They sound very similar, but they are talking about different things. For example, genotype is what? Again, purple. Versus white. Purple versus white. Genotype. What is a genotype? Again, big P, big P. So this is like the purebred parent. We call it homozygous dominant, okay? And then big P, small p, has to be italicized. Big P, small p, we call it heterozygous dominant. And then last but not least, small p and small p, we call it homozygous recessive, okay? So these are the three possible genotypes, but the ratio can be different. So for the next slide, I want you to identify the phenotype and genotype for the next slide. So hearing, you have a parent that are heterozygous dominant. So one parent is heterozygous dominant with big D, small d. The other parent is also heterozygous dominant with big D, small d. So if the parent has is homozygous recessive, then the offspring will if the offspring is homozygous recessive, then the offspring will be deaf. Otherwise, the dominant allele will give you the normal hearing. So the question to you is that what is the phenotypic ratio and what is the genotypic ratio? I'll give you 10 seconds to do this part. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, time's up. So basically, this is the same as as the Mendelian pea plant experiment. The phenotypic ratio. The phenotypic ratio, PR, is also 3:1. Because we have 3 hearing, normal hearing, but 1 deaf. That's why the phenotypic ratio is 3:1. What about the genotypic ratio? Genotypic ratio is also 1:1. Because we have 1 homozygous dominant, and then we have 2 heterozygous dominant, and we have 1 homozygous recessive. So the genotypic ratio is also 1:1. I hope you got it correct. Okay, monohybrid cross. We need to talk about that. So because I like to test students about monohybrid cross in the exam. So when in the exam, if I ask you about the result of certain cross, feel free to take out a piece of paper and then do the Punnett square. Write the Punnett square and then fill out the Punnett square. Then write out the phenotypic ratio and genotypic ratio. So yes, Mendel actually studied all 7 traits. one by one, and he got the same inheritance pattern. It's like they all follow the same rule. So the parental phenotype disappear in the F1 generation and then reappear 1/4 of the F2 offspring. So it's all the same. All 7 traits, they follow the same genetic rule. So this is a review. So the first one he studied is the flower color, and then he do the purebred. Basically repeat the same experiment. First of all, by using cell fertilization to get the purebred of the, let's say, flower position this time. So you got the purebred axial and purebred terminal, and then he do the crossing between parent, and they got the F1 generation. Then all the F1, they are axial, and then he crossed the F1 generation with each other. He got 3-to-1. So 3 axial and then 1 terminal in the F2 generation. And then he repeat the same experiment with the seed color, seed shape, pod shape, pod color, and stem length. And then he got the same 3-to-1 phenotypic ratio and 1-to-1 genotypic ratio, approximately. So he come up with this Mendels Law of segregation. Remember, we talked about segregation, that means separate before. It basically means that during meiosis, 2N becomes 1N. So all the daughter cell, all the gametes, they're 1N. So pairs of alleles segregate during gamete formation. Well, we learned this from meiosis, okay? And then fusion of the gamete fertilize at the fertilization create the pair allele again. Okay, let me tell you this in terms of math. So basically, this is the 2N. That means a pair of allele. They become 1N. 1N is the gamete. Now, when you have 2 gametes, the sperm fertilize an egg, you become a pair allele again. That means you become 2N. So basically, when we talk about chromosome number in meiosis or the diploid haploid, we are implying the allele as well, the pair of allele. So you may assume that the number of chromosomes, they correspond to the pairing of the allele. Okay, now we talk about homologous chromosome. Remember, homologous chromosome I mentioned in meiosis is that it's a pair of chromosome. That one chromosome comes from your father, one chromosome comes from your mother. Then we call homologous chromosome. And here give you a better definition: chromosome that carry alleles of the same genes. Okay, same gene, but they can be different allele. One allele can be dominant, one allele can be recessive. In this case, it will be heterozygous dominant. Or both allele can be dominant allele, then it will be homozygous dominant. The last case is that both allele may be recessive, then you will get homozygous recessive. A gene they are located in something called locus. Locus is basically a specific region of the gene along the chromosome. So if they are allele of the same gene, okay, they are located at the same locus. So same gene, they are located at the same locus. Different gene, they are located in a different locus. So it already said that they can they can carry identical allele or different allele. If they carry different allele, we call it homozygous dominant. If they carry identical allele, it can be homozygous dominant or homozygous recessive. Let me repeat, I may have said something wrong. Different allele. If the two chromosomes carry different allele, we call it heterozygous dominant. Heterozygous dominant. If they carry the identical allele, we call it homozygous dominant or homozygous recessive. So let's take a